the video. Chapter 2, The Brain. Disclaimer: I do not own the rights to the images or information provided. This was purely made from me to study for boards. The brain is composed of the embryologically segmented brainstem in two suprasegmental portions, the cerebrum, also known as the telencephalon, and the cerebellum, also known as the dorsometencephalon. The brainstem includes the myelencephalon, also known as the medulla oblongata, the ventral metencephalon, also known as pons, the mesencephalon, also known as midbrain, and the diencephalon, also known as thalamus, metathalamus, epithalamus, subthalamus, and hypothalamus. The cerebrum is divided into two cerebral hemispheres by the longitudinal cerebral fissure. Each cerebral hemisphere has outward folds, also known as convolutions, called gyri, and inward folds called sulci. Figures show the rostral and caudal parts of the lateral rhinal sulcus, the pseudosylvian fissure, the rostral and caudal sylvian gyri, the ectosylvian sulcus and gyrus, the suprasylvian sulcus and gyrus, the cruciate sulcus, the postcruciate and precruciate gyri, the coronal sulcus, the ectomarginal sulcus and gyrus, and the marginal sulcus and gyrus. Each cerebral hemisphere may be divided into lobes named for the portion of the calvaria that covers them. The relationship is not precise and varies among species. The frontal lobe is the portion of each hemisphere rostral to the cruciate sulcus. The precruciate gyrus is part of this lobe and functions as part of the motor cortex. The parietal lobe is caudal to the cruciate sulcus and is dorsal to the sylvian gyrus. It extends caudally to approximately the caudal third of the cerebral hemisphere. The postcruciate and rostral suprasylvian gyri are found in this lobe and function as part of the motor and sympathetic sensory cerebral cortex. The occipital lobe includes the caudal third of the cerebral hemisphere. Caudal portions of this lobe on both medial and lateral sides function as the visual cortex. The temporal lobe is composed of gyri and sulci on the ventrolateral aspect of the cerebral hemisphere. Parts of the sylvian gyri are located here and function as the auditory and vestibular system cortex. The rhinal sulcus separates the phylogenetically new cerebrum, also known as neopallium, dorsally, from the older olfactory cerebrum, also known as paleopallium, ventrally. Portions of the paleopallium that are visible are the olfactory bulb, which rests on the cribriform plate, and the olfactory peduncle, which joins the bulb to the cerebral hemisphere. On the next figure you can see, the olfactory peduncle courses caudally with a band of fibers on its ventral surface. Caudally, this band divides into lateral and medial olfactory tracts. The lateral olfactory tract passes caudally to the piriform lobe, which forms a ventral bulge just lateral to the pituitary gland and medial to the temporal lobe of the neopallium. On the next figure you can see the medial olfactory tract cannot be observed. On the medial side of the frontal lobe, facing the longitudinal cerebral fissure, are the frontal gyrus, the ectogenual sulcus, and the genual gyrus and sulcus. The callosal sulcus and cingulate gyrus are adjacent to the corpus callosum. The splenial sulcus and gyrus and occipital gyrus are the medial components of the occipital lobe. Each gyrus contains gray matter superficially and white matter in its center. The gray matter, or cerebral cortex of the neopallium, is composed of 6 layers of neuronal cell bodies. The white matter, the corona radiata, contains the processes, also known as axons, of neurons coursing to and from the overlying cortex. Next we discuss the cerebellum. The cerebellum is derived from the dorsal portion of the metencephalon and lies caudal to the cerebrum and dorsal to the fourth ventricle. The transverse cerebral fissure separates it from the cerebrum. The dural and osseous tentorium cerebelli are located in this fissure. On the next figure you can see the cerebellum is connected to the brainstem by 3 cerebellar peduncles on each side of the fourth ventricle and by portions of the roof of the fourth ventricle. The choroid plexus is a compact mass of pia, blood vessels, and ependyma. A choroid plexus develops when neural tube neuroepithelium did not proliferate to form parenchyma but remained as a single layer of ependymal neuroepithelial cells, referred to as a roof plate. More specifically, the layer of ependoma and the adjacent pia are referred to as the telechoroidea. The associated proliferation of capillaries is the choroid plexus. In common usage, all 3 components comprise the choroid plexus. These areas are found in the medulla, roof plate of the fourth ventricle, the diencephalon, roof plate of the third ventricle, and the telencephalon, roof plate of the lateral ventricle. At these sites, the vessels in the pia covering the single layer of ependymal cells proliferate to form a dense plexus of capillaries intimately related to these neuroepithelial cells. These cells and blood vessels are involved in passive and active secretion of CSF into the ventricular system. The choroid plexus of the fourth ventricle protrudes into the lumen of the fourth ventricle and is visible ventrolateral to the cerebellum, where it protrudes through the lateral aperture of the fourth ventricle on the lateral surface of the medulla. 3 cerebellar peduncles attach the cerebellum to the pons and medulla. The most lateral peduncle is the middle cerebellar peduncle, which continues to take the transverse fibers of the pons dorsocaudally into the cerebellum. The transverse fibers of the pons form a thick transverse band of fibers on the ventral surface of the pons. The caudal cerebellar peduncle extends dorsorostrally from the medulla into the cerebellum between the other two peduncles. The rostral cerebellar peduncle is the most medial on each side and primarily contains efferent axons from the cerebellum to the brainstem. Afferent axons to the cerebellum from the brainstem and spinal cord pass primarily through the middle and caudal cerebellar peduncles. The cerebellum is composed of lateral cerebellar hemispheres and a middle portion, the vermis. The convolutions of the cerebellum are known as folia. These are grouped into 3 lobes and numerous cerebellar lobules that have specific names. The vermis comprises the entire middle portion of the cerebellum directly dorsal to the 4th ventricle. Some of its lobules are found on the ventral surface of the cerebellum facing the roof plate of the 4th ventricle. Each hemisphere projects over the cerebellar peduncles and the adjacent brainstem. On the next figure you can see a lateral component lies in the cerebellar fossa on the medial side of the petrosal part of the temporal bone. The central portion of the cerebellum consists of a mass of white matter, the cerebellar medulla, which contains 3 pair of cerebellar nuclei, from medial to lateral. These are the fastigial, interposital, and lateral cerebellar nuclei. On the next figure you can see on the median section of the cerebellum through the vermis, the white matter shows a unique pattern as it branches and arborizes from the medulla of the cerebellum into the folia. This is referred to as the arbor vitae. Next we discuss the brainstem. On the next figure you can see, the internal capsule is the only structure connecting the cerebral hemisphere with the brainstem. It consists of neuronal axons projecting from the cerebral cortex to the brainstem and neuronal axons projecting from the thalamus to the cerebral cortex. The brainstem includes the diencephalon, mesencephalon, ventral metencephalon, also known as pons, and myelencephalon, also known as medulla. Diencephalon. The diencephalon consists of paired groups of nuclei separated by the centrally located 3rd ventricle and an adhesion. On each side is a large thalamus between the interthalamic adhesion and the internal capsule, as well as a smaller hypothalamus ventral to it, a lateral and dorsocaudal metathalamus, also known as the 2 geniculate bodies, a small ventrolateral subthalamus, and a very small hypothalamus on the dorsal midline. The diencephalon is the most rostral part of the brainstem. On the next figure you can see, on the ventral surface, the optic or second cranial nerves form the optic chiasm of the diencephalon rostral to the hypophysis, also known as pituitary gland. The optic tracts course laterally and dorsoquadrantally from the chiasm, pass over the lateral surface of the diencephalon, and enter the lateral geniculate nucleus of the metathalamus in this pathway. Each tract curves around the caudal edge of the internal capsule. Caudal to the optic chiasm on the median plane is the hypophysis, also known as pituitary gland, which is attached by the infundibulum to the tuber cinereum of the hypothalamus. If the gland is missing, the lumen of the infundibulum will be evident. This lumen communicates with the overlying 3rd ventricle of the diencephalon. The mammillary bodies of each hypothalamus bulge ventrally caudal to the tuber cinereum. They demarcate the most caudal extent of the hypothalamus on the ventral surface of the diencephalon. On the next figure you can see, the internal capsule bound to the diencephalon laterally. The thalamus, metathalamus, and epithalamus can be seen on the dorsal aspect of the diencephalon. 3 structures compose each epithalamus. They all are located adjacent to the median plane. 1. The stria habenularis lies on either side of the midline, coursing dorsally and caudally from the rostroventral aspect of the hypothalamus over the thalamus to the dorsicaudal aspect of the diencephalon. Here, the stria ensures the habenula nucleus. 2. The habenula nuclei on each side are connected by a small commissure. 3. Caudal to the habenula nucleus is the small, unpaired pineal body. This caudal projection from the diencephalon is small in the dog but very prominent in larger domestic animals. On the next figure you can see, a space is present between the stria habenularis of each side. This is the dorsal part of the 3rd ventricle. It is covered by a thin remnant of the roof plate of the neural tube, which is a layer of ependymit that extends from one stria habenularis to the other covered by a thin layer of pia mater. Branches of the caudal cerebral artery course over the diencephalon, and its capillaries form the choroid plexus of the 3rd ventricle. On the next figure you can see, rostrally, the choroid plexus of the 3rd ventricle is continuous with the choroid plexus of the lateral ventricle at the interventricular foramen. This foramen is caudal to the column of the fornix at the level of the rostral commissure. On the next figure you can see, the thalamus is bilateral, lying between the internal capsules on both sides and dorsal to the bilateral hypothalamus. It is covered by pia, arachnoid trabeculations, and the subarachnoid space. It consists of a multitude of ill-defined nuclei. Each metathalamus consists of the 2 geniculate nuclei, which are readily recognized on the caudal surface of the thalamus. A lateral eminence on the caudodorsal surface of the thalamus is the lateral geniculate nucleus, which receives axons from the optic tract and functions in the visual system. The lateral geniculate nucleus is connected with the rostral colliculus of the midbrain by a brachium. Quadventral to the lateral geniculate nucleus is the medial geniculate nucleus of the metathalamus. This nucleus functions in the auditory system and is connected to the caudal colliculus of the midbrain by the brachium of the caudal colliculus. On the next figure you can see, the interthalamic adhesion is an oval to round area of adhesion on the median plane between the left and right thalamic nuclei. It is surrounded by the third ventricle. In transverse section, the narrow vertically oriented third ventricle appears as a perpendicular slit ventral to the interthalamic adhesion. Its lateral and ventral walls are formed by the hypothalamus. The dorsal portion of the third ventricle is small and tubular. It passes dorsal to the interthalamic adhesion, where its thin roof plate is attached on each side to the stria habinularis. On the next figure you can see the mesencephalon. The mesencephalon, also known as midbrain, is relatively short. It is nearly round on transverse section with a canal, the mesencephalic aqueduct, passing through it. The mesencephalon consists of a tectum or roof dorsal to the aqueduct, which is composed of 4 groups of neuronal cell bodies, the caliculae. That portion of the mesencephalon ventral to the aqueducts is the cerebral peduncle on each side, which, from dorsal to ventral, consists of a tegmentum, also known as reticular formation, substantia nigra, ancra cerebri. On the next figure you can see, between the mammillary bodies of the hypothalamus and the transverse fibers of the pons is the ventral surface of the mesencephalon. The transverse fibers of the pons cover part of the caudal mesencephalon ventrally. Descending tracts of projection processes that connect portions of the cerebral cortex with brainstem centers in the spinal cord course on the ventral surface of the midbrain. These are grouped together on each side as the crus cerebri. The oculomotor, or third cranial nerve leaves the midbrain medial to the crus. On the next figure you can see the mesencephalic structures dorsal to the mesencephalic aqueducts compose the tectum of the midbrain. On the next figure you can see, the mesencephalic aqueduct is a short, narrow tube, derived from the neural canal in the midbrain, which connects the third ventricle rostrally with the fourth ventricle caudally. 4 dorsal bulges, the corpora quadrigemina, are evident on the dorsal side. On the next figure you can see, the rostral pair, the rostral colliculi, which function with the visual system. The smaller caudal pair, the caudal colliculi, which function in the auditory system. The trochlear, or fourth cranial, nerve courses laterally out of the roof of the fourth ventricle. The rostral medullary velum, adjacent to the caudal colliculus. It continues rostroventrally on the lateral surface of the midbrain. On the next figure you can see, the lateral lemniscus is a band of auditory system axons on the lateral side of the midbrain. It courses rostrodorsally from the level of the cochlear nucleus to the caudal colliculus and emerges medial to the middle cerebellar peduncle. Many of these fibers arise from the cochlear nucleus. The brachium of the caudal colliculus runs rostroventrally from the caudal colliculus to the medial geniculate nucleus of the thalamus. On the next figure you can see, on the dorsal surface, the commissure of the caudal colliculi can be seen crossing between these 2 structures. The rostral colliculus is connected to the lateral geniculate nucleus of the thalamus by a short brachium of the rostral colliculus. On the next figure you can see the ventral metencephalon. The metencephalic portion of the rhombencephalon includes ventrally a segment of the brainstem, the pons, and a dorsal development, the cerebellum. The ventral surface of the pons includes the transverse fibers of the pons, which course laterally into the middle cerebellar peduncles. This large band of transverse fibers borders the trapezoid body of the medulla caudale. Its rostral border covers part of the ventral surface of the midbrain. The trigeminal nerve is associated with the pons, entering it along the quadrilateral aspect of the transverse fibers. The caudally projecting axons of the crus cerebri enter the pons dorsal to the transverse fibers, where they are called the longitudinal fibers of the pons. These longitudinal fibers, also known as axons, are covered ventrally by the transverse fibers. The longitudinal fibers that do not terminate in pontine nuclei continue caudally on the ventral surface of the trapezoid body of the medulla as the pyramids. Many of the axons in the crus cerebri and the longitudinal fibers of the pons and most of those in the transverse fibers of the pons make up a large cerebropontocerebellar pathway. Synapse occurs in the pontine nuclei that are covered by the transverse fibers, and crossing occurs through the transverse fibers to enter the cerebellum through the contralateral middle cerebellar peduncle. Therefore, impulses that arise in the left cerebral hemisphere are projected to the right cerebellar hemisphere. The rostral medullary velum forms the roof of the fourth ventricle between the caudal colliculi of the mesencephalon rostrally and the midventral surface of the cerebellum caudale. As seen on the next figure, the crossing fibers of the trochlear nerves course through this velum. The velum lies over the floor of the fourth ventricle and loosely covers the caudal opening of the mesencephalic aqueducts into the fourth ventricle. This velum lies between the fourth ventricle, which it covers, and the rostral cerebellar vermis. Between the velum and the vermis is the subarachnoid space. On the next figure we discuss the myelencephalon. The myelencephalon, or medulla oblongata, extends from the transverse fibers of the pons to the level of the ventral rootlets of the first cervical spinal nerve. The trapezoid body is the transverse band of fibers rostrally that course parallel but caudal to the transverse pontine fibers. It is continuous with the vestibulocochlear nerve and cochlear nuclei laterally on the side of the medulla and functions in the auditory system. The pyramids are a pair of longitudinally coursing bundles of axons on either side of the ventral median fissure. They emerge from the transverse pontine fibers as the caudal continuous shines of axons from the longitudinal fibers of the pons that did not terminate in pontine nuclei. They course caudally across the trapezoid body to continue on the ventral surface of the medulla. They are separated by the ventral median fissure, which can be followed caudally until it is obliterated over a short distance by the decussation of the pyramids located at the level of the emerging hypoglossal nerve axons. The decussation itself is indistinct because it occurs as the pyramidal axons are passing dorsally into the parenchyma of the medulla. Pyramidal axons continue in the spinal cord as the corticospinal tracts. These are cerebral projection axons that project to the spinal cord. On the next image you can see, the abducent, or sixth cranial, nerve leaves the medulla through the trapezoid body on the lateral border of each pyramid. Cranial nerve 7, the facial nerve, is smaller and leaves the lateral surface of the medulla through the trapezoid body caudal to the trigeminal nerve and just ventromedial to the eighth cranial nerve. On the next image you can see the nuclei of cranial nerve 8, the vestibulocochlear nerve, is on the lateral side of the medulla at the most lateral extent of the trapezoid body just lateral and dorsal to the facial nerve. The cochlear nuclei are located in this nerve, where it attaches to the medulla. All cochlear neurons in the vestibulocochlear nerve synapse cochlear nuclei. The axons of these cell bodies in the cochlear nuclei continue into the medulla by one of two pathways. Some continue as the acoustic stria and pass over the dorsolateral surface of the medulla and caudal cerebellar peduncle to enter the medulla. Others pass on the ventral surface in the trapezoid body. The vestibular neurons in the vestibulocochlear nerve pass directly into the medulla, where most of them synapse in a vestibular nucleus. On the next image you can see, the hypoglossal nerve, cranial nerve 12, emerges as a number of fine rootlets from the ventrolateral surface of the myelencephalon caudal to the trapezoid body, lateral to the caudal portion of the pyramid. The junction of the myelencephalon and the spinal cord is between the hypoglossal axons and the ventral rootlets of the first cervical spinal nerve. The hypoglossal nerve outside the skull is much larger, owing to the addition of connective tissue components. Dorsolateral to the emergence of the hypoglossal axons and the axons of the ventral root of the first cervical spinal nerve, a nerve runs lengthwise along the lateral surface of the spinal cord. This is cranial nerve 11, the accessory nerve. Its spinal rootlets emerge from the lateral surface of the spinal cord as far caudally as the seventh cervical segment. They emerge between the level of the dorsal and ventral rootlets of the cervical spinal nerves and course cranially within the subarachnoid space adjacent to the spinal cord through the vertebral canal and foramen magnum. These spinal rootlets form the external branch of the accessory nerve. A few cranial rootlets emerge from the lateral side of the medulla caudal to the 10th cranial nerve, form the internal branch, and join with the external branch to form the accessory nerve. The accessory nerve leaves the cranial cavity through the jugular foramen and tympano-occipital fissure along with the 9th and 10th cranial nerves. As these nerves traverse this fissure, the internal branch components of the accessory nerve join with the vagus nerve for distribution to the esophagus and the larynx. Cranial nerves 9 and 10, the glossopharyngeal and vagal nerves, comprise small rootlets on the lateral side of the myelencephalon caudal to the 8th cranial nerve and rostral to the accessory nerve. On the next figure you can see, when the cerebellum is removed from the brainstem, the cut ends of the three cerebellar peduncles are visible on either side of the fourth ventricle. The rostral cerebellar peduncle is medial and courses rostrally into the mesencephalon. The middle cerebellar peduncle is lateral and arises from the transverse fibers on the lateral side of the pons, and the caudal cerebellar peduncle is in the middle, entering from the myelencephalon after passing ventral to the acoustic stria. The dorsal surface of the pons and medulla is covered from rostral to caudal by the rostral medullary velum, the cerebellum, and the caudal medullary velum. This covered space is the fourth ventricle. The groove in the center of the floor of the fourth ventricle is the median sulcus. On the lateral wall, the longitudinal groove is the sulcus limitans. Just lateral to this sulcus, at the level of the acoustic stria, a slight dorsal bulge of the medulla is present. This demarcates the location of the vestibular nuclei. Most of the vestibular neurons in the vestibulocochlear nerve terminate here. The roof of the fourth ventricle caudal to the cerebellum is the caudal medullary velum. It is a thin layer composed of ependyma lining the ventricle and a supporting layer of vascularized pia. It attaches to the cerebellum rostrally. This attachment is in the midventral portion of the cerebellum between the rostral and caudal ventral portions of the vermis. This velum attaches to the caudal cerebellar peduncle and the fasciculus gracilis laterally and caudally. Its attachment caudally at the apex is known as the obex. At this level, the fourth ventricle is continuous with the central canal of the spinal cord. At the level of the 8th cranial nerve, dorsal to the acoustic stria, is an opening in the caudal medullary velum. known as the lateral aperture of the fourth ventricle. The CSF produced in the ventricular system communicates with the subarachnoid space of the meninges via this aperture. It then courses through the subarachnoid space over the entire surface of the brain and spinal cord and is absorbed into the venous system. Most of this absorption occurs where the arachnoid is in close opposition to the cerebral venous sinuses and where it has formed specialized structures known as arachnoid villi. CSF is also absorbed from the subarachnoid space, where the spinal nerves leave the vertebral canal through the intervertebral foramina, and along the olfactory and optic nerves. The choroid plexus of the fourth ventricle bulges into the lumen of the ventricle on each side of the dorsal midline. Rostrally, where the caudal medullary velum attaches to the ventral surface of the cerebellum, each choroid plexus extends laterally through the lateral recess and aperture, where it can be seen ventral to the cerebellum. On the next image you can see the median groove on the dorsal surface of the myelencephalon. Caudal to the fourth ventricle is the dorsal median sulcus. The narrow longitudinal bulge flanking the sulcus is the fasciculus gracilis. This longitudinal tract projects cranially the entire length of the spinal cord in this position. At the caudal end of the myelencephalon, it ends at the nucleus gracilis. This nucleus is located at the caudal end of the fourth ventricle, where the fasciculus widens and ends. This fasciculus and nucleus function primarily in the general proprioception of the pelvic limb. The groove lateral to the fasciculus gracilis is the dorsal intermediate sulcus. The longitudinal bulge lateral to this is the fasciculus cuneatus. This tract also projects cranially on the dorsal aspect of the spinal cord, starting in the mid-thoracic region. The fasciculus cuneatus diverges laterally at the caudal end of the fourth ventricle and ends in a slight bulge. This bulge represents the lateral cuneate nucleus and is known as the cuneate tubercle. Rostrally, the lateral cuneate nucleus is continuous with the caudal cerebellar peduncle. This fasciculus and nucleus primarily function in the general proprioception of the thoracic limb. On the next image you can see, the groove on the caudodorsal surface of the myelencephalon lateral to the fasciculus cuneatus is the dorsolateral sulcus. The longitudinal bulge lateral to it is the spinal tract of the trigeminal nerve. The axons in this tract are from sensory neuronal cell bodies in the trigeminal ganglion that supply the head. The trigeminal tract extends quadrantally to the level of the first cervical segment of the spinal cord because of the large number of cell bodies serving its extensive peripheral distribution in the head. This tract emerges on the lateral surface of the myelencephalon caudal to a band of obliquely coursing ventral to dorsal superficial arcuate fibers. These arcuate fibers, also known as axons, connect structures in the medulla with the caudal cerebellar peduncle. The dorsal rootlets of the spinal cord enter it through the dorsolateral sulcus that extends along the spinal cord. Telencephalon, cerebrum. One cerebral hemisphere can be removed by cutting the rostral commissure, corpus callosum, and hippocampal commissure, separating the halves of the body of the fornix on the median plane, and sectioning the internal capsule, which is the sole structure attaching the cerebral hemisphere to the thalamus of the brainstem. 3 commissural pathways cross between the hemispheres, one for each phylogenetic division of the cerebrum. The corpus callosum connects the neopial portion of each hemisphere and is the largest of the 3 pathways. The rostral commissure connects the paliopial or olfactory components of each hemisphere. On each side, this commissure connects rostrally to the olfactory peduncles and bulbs and caudally to the piriform lobes. The hippocampal commissure is small and is located just caudal to the junction of the crus of each fornix to form the body of the fornix. This commissure connects the archipodial components of each hemisphere. The large internal capsule consists of projection axons that course between the cerebral hemisphere and the brainstem. Association axons remain within the hemisphere, coursing between the adjacent or distant gyri. The corpus callosum consists of a rostral genu, the middle body, and the caudal splenium. It is the commissural pathway for axons crossing between the neopallium of each cerebral hemisphere. On the next figure you can see, the internal capsule contains projection axons that course between the telencephalon and the diencephalon and project caudally from the telencephalon to the brainstem and the spinal cord. The axons that were previously seen in the mesencephalon as the crus cerebri came through the internal capsule. The projection pathway of most sensory modalities to the cerebrum for conscious perception involves synapses in the thalamus. The axons of these thalamic cell bodies then enter the internal capsule to reach the cerebrum through the thalamicortical axons. On the medial surface of the cerebral hemisphere, a thin vertical sheet of tissue extends between the corpus callosum and the fornix. This is the septum pellucidum, which is more developed rostrally where it extends from the genu to the rostral commissure. A thickening in the septum dorsal and rostral to the rostral commissure represents the septal nuclei. Caudal to the rostral commissure, the septum attaches the corpus callosum to a column of axons that courses rostrally and then turns in a rostroventral curve primarily caudal to the rostral commissure. On the next figure you can see, these axons are part of the fornix. They connect the hippocampus with the diencephalon and the rostral cerebrum. The hippocampus is a specialized region of cerebral cortex that forms an internal gyrus located in the wall of the lateral ventricle. The fornix begins caudally by the accumulation of axons, called fimbria, on the lateral side of the hippocampus. The fimbria coalesce to form the crus of the fornix. The crura from each hippocampus join rostral to each hippocampus and dorsal to the thalamus to form the body of the fornix. The body of the fornix courses rostrally and then curves rostroventrally as the two columns of the fornix. At the rostral commissure, some axons course dorsal and rostral to the rostral commissure, but most continue ventrally caudal to the commissure and continue cauda-ventrally, lateral to the third ventricle, to reach the mammillary body of the hypothalamus. The rostral commissure develops in a thin sheet of neural tube, the lamina terminalis. This is the most rostral extent of the diencephalon of the brainstem and represents where, in development, the two telencephalic vesicles grew out of the prosencephalon. The curved cavity lateral to the septum pellucidum and ventral to the corpus callosum is the lateral ventricle. It communicates with the third ventricle of the diencephalon by the interventricular foramen, which is located caudal to the column of the fornix at the level of the rostral commissure. The quadradorsal wall of this foramen is the roof plate and choroid plexus of the third ventricle. The lateral ventricle is C-shaped, with its ventral portion in the temporal lobe just caudal to the amygdala, which is a basal nucleus in the piriform lobe. This ventricle can be considered to start at the caudal extent of the piriform lobe and curve dorsocaudally and then rostrally and ventrally to end at the interventricular foramen. The hippocampus and its fornix have the same curved shape as they lie in the wall of this ventricle. Phylogenetically, the cerebral cortex can be divided into three regions: piliopallium, neopallium, and archipallium. The piliopallium consists of the cortex of the olfactory peduncle. This is separated laterally from the neopallium by the rhinal sulcus. The neopallium includes all of the gyri on the external surface of the cerebrum dorsal to the rhinal sulcus. The hippocampus belongs to the archipallium and is an internal gyrus of the telencephalon that has been rolled into the lateral ventricle by the lateral expansion of the neopallium. The hippocampus begins ventrally in the temporal lobe and curves, first caudodorsally and then rostrodorsally and medially, over the diencephalon to reach the middle of its caudodorsal aspect. At that point, the hippocampus ends, and a hippocampal commissure connects the hippocampus of each cerebral hemisphere. The axons of the hippocampus form the fimbria on its surface that accumulate on its lateral side as the crus of the fornix. The two crura join at the midline dorsal to the diencephalon to form the body of the fornix. The body continues rostrally and then curves ventrally to form two columns of the fornix at the level of the rostral commissure. Attached to the lateral free edge of the crus of the fornix is a network of blood vessels covered by arachnoid trabeculae, pia, and ependyma. This is the choroid plexus of the lateral ventricle. Its anatomic structure is the same as the choroid plexus of the third and fourth ventricles. The surface of the choroid plexus that faces the lumen of the ventricle is the ependymal layer derived from the embryonic neural tube. This layer of ependoma and pia is attached on one side to the free edge of the fornix. To complete the wall of the lumen of the lateral ventricle, it must be attached on its other side. On the next figure you can see, this attachment is to a small tract, the stria terminalis, located in the groove between the thalamus and the caudate nucleus. The stria terminalis is a small tract that connects the amygdala with the septal nuclei. This layer of ependoma from the stria terminalis to the fornix forms part of the medial wall of the lateral ventricle. Branches of the middle and caudal cerebral arteries covered by pia push this layer of ependoma into the lumen of the lateral ventricle. The result is the formation of a choroid plexus that is continuous with the choroid plexus of the third ventricle at the interventricular foramen. On the next figure you can see in each cerebral hemisphere, the neuronal cell bodies are located either on the surface in the cerebral cortex or deep to the surface in a basal nucleus. The caudate nucleus is a basal nucleus that forms part of the wall of the lateral ventricle and has a similar C-shaped curve. On the next figure you can see, rostrally, its large head bulges into the floor of the lateral ventricle. Its body narrows as it courses caudally and continues into its narrow tail. On the previous image you can see, the latter curves ventrally and then rostrally to terminate at the amygdaloid body, a basal nucleus in the piriform lobe. Thus, the head and body of the caudate nucleus are on the floor of the dorsal part of the lateral ventricle, and the tail is in the roof of the quadventral part of the lateral ventricle. Other basal nuclei in the frontal lobe of the cerebrum include the lentiform nucleus, which is ventrolateral to the rostral portion of the internal capsule. Its two components are the putamen laterally and the globus pallidus medially. The external capsule bound to the lateral portion of the putamen. The nucleus accumbens is located ventral to the head of the caudate nucleus. The endopeduncular nucleus is part of the subthalamus located primarily between the ventral portion of the internal capsule and the optic tract. The claustrum is a narrow band of gray matter lateral to the external capsule. On the next image you can see, dorsolateral to the caudate nucleus, the internal capsule forms the lateral angle of the lateral ventricle. At the dorsolateral angle of the lateral ventricle, the axons of the internal capsule meet those of the corpus callosum. Arising from this interdigitation of axons, the corona radiata radiates in all directions to reach the gray matter of the cerebral cortex. A longitudinal section of the cerebral hemisphere in the dorsal plane at the level of this interdigitation reveals a mass of white matter in the center of the hemisphere. This mass is referred to as the centrum semiovalle. Functional and structural correlates of the nervous system are listed in Table 2-1.